Yeah, good still morning, everybody. First of all, I would like to thank the organizers here of the Humboldt Kolleg, the Humboldt Foundation, and especially Professor Mugesh, who is here my local host, and we have already collaborated and met when he was a Humboldt Fellow in Germany, and since that time we have quite successful and productive cooperation, which recently even led to a joint publication, which I'm very pleased about. So today I will introduce you into a topic of human health, and human health related to thyroid hormones, and you will see that it's a rather interdisciplinary approach which we are following over the last few years, because we are working on hormones. That is our the mission in the Institute of Experimental Endocrinology, which is at the Charité in Berlin. And Charité is the joint medical faculty of Humboldt University and Free University. I think this is a unique situation in the world, that a medical faculty belongs to two different universities which are competing. This is a political decision by our local state government. So I'm nominated and called as Humboldt professor, and I'm cooperating and competing with my Free University professors in the same medical faculty. I think this is quite interesting. This experience goes now for 6-7 years, and we'll see how it works out if it's also used somewhere else. Now, we have received funding from various sources, and you have seen already major funding sources, the German Research Foundation, DFG. I have had DAAD fellows in my laboratory, both before in Würzburg and in Berlin now, and we also have good networks with Humboldt fellows. So I think there is a lot of resources available, and you just have to try, you just have to look for where is the funding, which program fits to my purposes, and you have to get some colleagues cooperating, and then you will have chances really to get what you want. Now, the topics will be, and this is on the next slide, trace elements, endocrine disruptors at the end, how they interfere with thyroid hormones in general. So we will cover chemistry, biochemistry, inorganic chemistry. We will cover modern concepts of hormone transport, synthesis and action. We will target a few specific proteins which are very peculiar, like a peroxidase, thyroid peroxidase, which is essential for the hormone synthesis, and a group of enzymes which is very unusual because they are based on selenocysteine in their active site. And most of you know that the genetic code, since it was discovered initially, has been expanded. Meanwhile, there are 22 proteinogenic codons. Many textbooks still are 20 years behind because they call there are only 20 proteinogenic codons, but there are 21 at least. One is selenocysteine, and the second one which is coding for pyrolysin. So we will then go into structural aspects of a hormone binding site or a ligand binding site, and finally to its exploitation as a target for drugs. And of course, finally at the end we have to deal also with our environment, which is interfering among others with the hormone system. So why do we actually work on the thyroid hormone axis? Why am I interested in that? It is well known to you that thyroid hormone effects are essential for development, and especially brain development in human beings and also in most of the animals and higher vertebrates. And we have still a major problem in the field, that we lack worldwide iodine as an essential trace element. When it comes to disease, 1/3 of females during their reproductive age or menopause can be affected by a disease which is called autoimmune thyroid disease, morbus Hashimoto, thyroiditis, and this is the reason why thyroxine, the synthetic hormone, since more than 90 years now, has been used as a drug, and in Germany this is drug number 3 prescribed in pills. Worldwide it's among the 10 most prescribed drugs. It's not an expensive drug. It doesn't make a big market, but it is used in many, many individuals, and especially females. And when it comes now to the worldwide problem of obesity, we know that thyroid hormone is one of the most potent hormones regulating energy metabolism and obesity. So there is a major target to get your thyroid status right in order to prevent obesity adipositas with all the sequelae of diseases. When it comes now to cancer, also a worldwide problem. Among the endocrine cancers, thyroid is leading as the most frequent cancer. So we have in Germany a rate of 5 to 7 per 100,000 of cancers. So it's among the endocrine cancers the most prevalent. And of course, it is quite interesting now because recently we learned that there is more than one thyroid hormone. You know in the textbook there is one T3-Alcampotec, but we know there is much more to that. And as we know, the population worldwide is vulnerable to these changes and dysfunctions of the hormone system. So I think it's a very powerful and interesting part. Now, what do thyroid hormones do? I don't go into the details of endocrinology, but they are essential for neurodevelopment. They are essential for adequate growth of humans and animals. They are essential for metabolism, not only energy metabolism, but also all intermediate metabolism, structural metabolism, bone metabolism. I do not know one biological system in humans or animals which is not affected by thyroid hormones. So they are not very specific, but they touch everything. They modulate everything by this issue. And among the most important ones are, of course, body temperature and body composition. So thyroid hormones are highly thermogenic. They increase body temperature and, of course, then energy turnover. And they affect specific genes, proteins, and enzymes. And for those who are interested in history a little bit, I was learning here on the famous Professor Tata. When it comes to hormone action, there is another Tata who is still alive, Yamachet Tata, who is living in UK. And he was the first one to show with thyroid hormone that gene expression is affected 60 years ago. So I think it's a rather archaic system, and in order to give you a little bit of an idea of how complicated thyroid hormone biosynthesis is, I will not go into details. We need, first of all, the essential trace element iodine. And then we need to oxidize iodine to a state which is called iodine 0 or iodine plus. And to do that, you need an enzyme, of course, which is thyroid peroxidase, TPO, and TPO generates H2O2. And everybody in the room knows that H2O2 is toxic. So to generate thyroid hormone, you have to devise a system which is generating toxicity lifelong, from the 12th week of pregnancy until you die. You generate a cytotoxic agent H2O2, and this is a problem, of course. If you have too much H2O2, you would burn, you would inactivate your thyroid gland. And that's why you need an antioxidative defense system like glutathione peroxidase, which can reduce H2O2 to generate water. And that's why you use then another trace element. To make the synthesis now, it's not as you would do it now if you are a chemist. You would design, I take a precursor and make the product by modification. No, it's a very complicated way. You make a huge protein, 2 times 330,000 molecular mass, which you deposit outside of the cell, and then you do solid phase chemistry on this extracellular protein. If we would reinvent the system, nobody would design that as nature did it over the years. Now, with this big protein, you can incorporate iodine, which you have oxidized, and then you modify this protein. I come to the details, and finally there comes some thyroid hormone out. Thyroxine, T4 with 4 iodine atoms, T3, the active form with 3 iodine atoms. But that's not all. We have learned over the last 10 years there are several, 2 more at least, new thyroid hormones. One is a biogenic amine called 3T1AM, which has been rediscovered. We do not know really how it acts, but what we know, it is antagonizing the biological active hormone in that way. And we do not know if it's made directly by the thyroid one hypothesis, or if it's made from these precursors outside of the body of the thyroid gland. So these are open questions which we are studying. So to summarize, we need iodine, we need iron, this is a hemoprotein, the peroxidase, we need selenium as a defense system, and we need also later on zinc as a force hormone. Now, these are the explanations shortly for these abbreviations, and we know now how thyroid hormone is made theoretically. But where is it made? It's made in the human thyroid gland. Everybody has it, you think. No, not everybody has it. 1 in 3,000 individuals worldwide does not have a functioning thyroid gland, but they are alive. Why are they alive? Because in the 80s, a screening system was introduced that every newborn worldwide now is screened for thyroid function, and if it's not there, these individuals have to get thyroxine in their first week of life, and they will be happy until to the end of their life as long as they take thyroxine. So you can live without a thyroid gland, but you cannot live without thyroid hormone. Adequate life. Now, where is this made? So this is a very complicated structure. You see here called an epithelial structure, magnified here. And every of these cells of this thyroid follicles, what we call it, the follicular unit, has access to a microcapillary. So blood is coming to every individual thyrocyte. There are more endothelial cells in the thyroid than thyrocytes which are producing the hormone. This is completely neglected in most textbooks. Nobody talks about the vascular system. But without vascular system, you cannot bring the iodine to the gland, and without a vascular system, you cannot export the hormone which is made by the gland. So you need vascular access. And now, where is it made, the hormone? The hormone is not made in the cell, which is also written in many textbooks, but it's wrong. The hormone is made in this lumen, in this colloid hole. So there, this thyroglobulin is produced, secreted, disposed there, stored there, and then extracellular, not intracellular, all these biochemical reactions occur, which I mentioned before. So this is extracellular chemical synthesis, not intracellular hormone production. And then, by chance, when you want and need, you take this thyroglobulin, you take it up in the cell, you modify it, you hydrolyze it completely with a lysosome, and then you can deliver your hormone with to the capillary system. So more complicated than you would invent it if you would make it new. Now, this is kind of a summary cartoon. I do not go in the details. You have to take up iodide with a supporter which is not electroneutral. You immediately transport the iodide out into the colloid lumen. There you then find the enzyme which is generating H2O2, a dual oxidase, and the thyroid peroxidase. The hemoprotein is using H2O2 to oxidize iodine, to iodinate it, to couple it to thyroid hormone. You store it, and then you can, by chance, take it in and generate with lysosomal proteolysis the hormones. But how is that regulated? Everything is regulated in the thyroid world with a TSH receptor. And this signal comes as a pituitary hormone to the thyroid gland and generates how much thyroid do I synthesize, how much thyroid do I release. So this is under a classical endocrine feedback system. So we are ready to go. So you have to solve this problem, H2O2 defense. You have to store the hormone in the iodinated thyroglobulin, and you have a TSH-regulated synthesis, storage, and release. This has to work, and you have a local reservoir of trace elements, iodine, selenium, and iron for this purpose. Now, of course, TPO is known. Can we interfere with thyroid hormone synthesis? And all medication which is interfering with thyroid hormone function is currently based on an interaction with the thyroid hormone synthesizing enzyme TPO, which is a hemoprotein, and there are only 2 drugs available to treat hyperthyroidism, too much thyroid hormone, and these were invented in the Second World War and used until today. They have a lot of side effects, and pharmaceutical industry is not interested in developing new drugs. So we urgently need new drugs for thyroid hormones, and especially in the cooperation with Dr. Mugesh, we try to address this issue to get new targets available and new structures which can interfere with thyroid hormone biosynthesis. So these are the two drugs, propylthiouracil, methimazole. Now, this would complete the cartoon. I don't go into details. So we have a whole set of synthesis, and then we have a whole set of enzymes and proteins protecting the thyroid gland from H2O2. So there is an own world of protection. This is the reason why thyroid hormone has so high selenium contents, to defend against this H2O2 by addressing glutathione peroxidases, producing thyroidoxin reductases, control the redox state of the cells. So you need urgently selenium to make thyroid hormone and to keep your thyroid working. So iodine deficiency is not the only reason of malfunction of a thyroid. It can also be selenium deficiency. And the third reason of malfunction of the thyroid can be iron deficiency, neglected. Because you have to have this thyroid peroxidase, the hemoprotein, and you need iron, and many of the problems of worldwide inadequate synthesis of thyroid hormone are impaired by inadequate iron intake. So one of the reasons why females might be more susceptible to thyroid diseases could be that they lose, during their reproductive age, via menstrual cycle, blood and are iron inadequately supplied. So three reasons could be involved there. Now let's go to these trace elements. You know the chemical structure: selenium, iodine in the halogen group, iron and zinc. So let's talk a little bit on selenium. When do we know what happened to selenium? It was discovered a long time ago by this guy, Jöns Jakob Berzelius in Sweden, 1817. And it took more than 140 years until it was recognized that selenium is an essential trace element for human and many animal life forms. Plants don't use it. Selenium was then identified as a constitutive constitution of glutathione peroxidase, and in 1990 our group and a group in Scotland discovered that the deiodinases are selenocysteine containing enzymes. So that's how I came in that field and stayed to that because it was so exciting. Now what happened to selenium after it was discovered? It was known that it's a highly toxic compound. It was labeled as a carcinogen. And then in 1957 it was an essential trace element. In 1966 it was shown that it's even an anti-carcinogen if you use it correctly. And then in 1990, when we then discovered that it's involved in the selenium regulation of thyroid hormone synthesis and degradation, you see the picture changed dramatically and this guy left the bottle. Still, selenium is toxic if you take too much, but it's essential also. Now, why is it essential in humans? There are two protein groups which contain selenium. You have one group, selenocysteine containing, where the sulfur, typically of cysteine, is replaced by selenium. And this, of course, changes chemical characteristics, makes it more specific, more nucleophilic, and so on, and more reactive. And the interesting part is to make this amino acid part of a protein, you need the 21st proteinogenic codon. You cannot just use the code for cysteine. You need to reprogram the whole ribosome to make these proteins containing selenocysteine. A second version of selenium proteins comes in selenomethionine. Here the situation is different. The nature enzymes, biosynthesis does not discriminate between sulfur methionine and selenium methionine. So you can randomly incorporate selenium into selenomethionine and use it in the protein biosynthesis. Of course, structural chemists know that they can dotate then their proteins to get more signal in their X-ray diffraction. But here is no discrimination. But this is also a chance for nature, because if you store selenium in selenomethionine, you can improve your selenium store. You can avoid deficiency. But you cannot use that for any specific function. You cannot take this selenium and replace it here. You have to go to the degradation, to the molecule, to the element, and then with the element you can come back with selenocysteine. So it's rather complicated to utilize this selenium. And as we have learned over the last years, the world of selenoproteins, we are just here somewhere at the iceberg. We know a few enzymes with their function. Then in 2003, the group of Gladyshev discovered that the human genome contains 25 genes coding for selenocysteine-containing proteins. And we know now that the selenoproteome, out of these 25 genes, covers something like 30 to 70 selenoforms, protein forms. Different translational start site, alternative splicing, and so on and so on. And since 2005, we know this can go wrong. According to Murphy's law, what can go wrong will go wrong. And here we have also, of course, mutations leading to impaired selenoprotein biosynthesis and synthesis. And lo and behold, how were they discovered? The first discovery of this mutation of selenocysteine binding protein 2 was because the thyroid function of these patients is impaired. So the first signature of a severe selenium deficiency or a genetic deficiency shows up clinically in the thyroid system. They have inadequate thyroid hormones. So it seems to be relevant. Now, how does this happen? And I don't go to the system. To reprogram a UGA, the 21st codon, encoding selenocysteine into the protein, you need various types of factors. You need a specific tRNA, you need a specific elongation factor replacing the normal elongation factor, and then you need a protein which is called thesis binding protein 2, which recognizes a specific hairpin structure of the trans mRNA encoding selenocysteine containing proteins, and with this complex intact, you then can replace what would typically be read as a stop codon and incorporate in this context selenocysteine into a nascent protein chain. Very complicated, highly regulated, slow, error-prone, but it works. And if this protein is not working, then you run into trouble, especially with all other selenoprotein biosynthesis, which will be impaired and, of course, affected. Now, how do you integrate your selenium into your biological system? You have various chances. You can take up selenomethionine from plant or meat, which is not regulated. You can have then a store, or you can then use selenocysteine with this complicated mechanism, which is described here, make selenoproteins. One is a very specific one, a storage and binding proteins secreted by the liver. And then you have always, for this system, you always have to go to a reduced form like HSE minus to use it into the selenocysteine protein. If you have too much selenium, you can incorporate it into sugar, or you can methylate this, hydrogen selenide and breathe it out or lose it in the urine. So there is a kind of protection against toxicity, and various forms of selenium compounds can be used and absorbed in the intestine, and in typical nutrients you find selenomethionine, or you can use selenite or selenate as a nutritional supplement. So that's how you get it. What happens if you get too much selenium? This happens, and I show you just a picture here out of India. From the textbooks you can get, sorry for the wording, loose hair and nails. You shed off the nails, and you get also problems with bone development similar to rachitis, vitamin D deficiency. So it is a very complex picture of intoxication, so you should avoid intoxication. And this is, of course, not a new message. It was already told by Paracelsus, omnia sunt venena, nihil est sine veneno, sola dosis facit venenum, the dose makes the poison. Of course, known since many, many years. This also is valid for selenium. So where should we go with our selenium? The German nutrition recommends 30 to 70 micrograms, Nordic recommendation approximately 50 micrograms, and currently we think that if you exceed 300 microgram intake chronically over time, then you will run into problems of toxicity. If you just have a single dose of that, it doesn't matter, but if you have chronic exposure, it's really a problem. And there is a discussion recently that if you get too high intake of selenium chronically, you might increase your type 2 diabetes risk. But I do not believe that story, because I think it's artifact of statistics. There is no good proof at this point from basic science, so we have to work on that and see where it goes to. Now, can you measure what your selenium status is? Yes, you can. And we and others have developed an immunoassay for one of the selenoproteins, which is called selenoprotein P, and this can be used as a selenium marker. Why can it be used? Because this is the only protein which we know which contains in one peptide chain 10 times in a human selenocysteine. So to make this protein, the ribosome has to work really very, very hard. 10 times reprogramming, exchange of the translation factors, and so we do not know yet how it works, but it works. Liver secretes this protein, it stores this protein, and this selenoprotein is then used by other tissues like testes, like kidney, like the brain, via a receptor-mediated uptake of this protein, which is then locally proteolyzed, probably delivering selenium to the target tissues. So, long story to get your selenium status correct. And we have data from the literature showing that if your intake of selenium is approximately equal to 150 micrograms, up to 200 micrograms per day, you kind of saturate this system and you can come to an adequate selenium status. So that's why we think it's a good biomarker for the selenium status. What about selenium deficiency? Interestingly, you saw that selenium excess, for example, is leading to a clinical picture affecting bone development. And if you see selenium deficiency, also bone development is affected. So the toxicity and the deficiency kind of tag the same target in the bone, the chondrocyte. And there is a disease called Cushing-Beck disease, or big joint disease, where chondrocyte development is not proper. These people stay short and have problems with their joints, as you see, and are, of course, affected by other kinds of deficiency. There is a second disease called Keshan disease, which mainly affects myocard function, again, more in females, especially also lactating and pregnant females than in the non-pregnant ones. So, nevertheless, if there is toxicity and deficiency, is selenium already in the clinical practice? Yes, it is. It is used in prevention of goiter and treatment of autoimmune thyroid disease, both forms, the autoimmune thyroiditis and Graves' disease. Because of this protection for the thyroid gland, probably, we do not understand the mechanisms. There have been very good studies, still controversial, that adequate selenium intake will reduce cancer incidence and mortality, especially for prostate and colorectal cancer. No evidence for breast cancer or other ones, or brain meningioma. So there has been a lot of work going on. If you have nutritional imbalances like short bowel syndrome, also you have to replace trace elements, among also selenium, and especially in another hereditary disease of phenylketonuria, where you have to increase your protein intake, you have to replenish selenium then to make it going on. And I mentioned already, too high selenium intake, though you should not overdose it, might give you a risk for toxicity and also for type 2 diabetes. Now, the other trace element, iodine. I mentioned initially that iodine is so important for the gland. Yes, it is. And if you see what's happening to iodine, 1.6 billion of our population on this globe are still under the risk of inadequate iodine intake and impaired mental development. We could increase our worldwide IQ by 10 to 15% with adequate iodine intake. It did not happen. WHO wanted to do that by year 2000. Year 2000 has passed since 14 years, and we still have iodine deficiency worldwide. What are the consequences? We have 600 million of people with goiter, enlarged thyroid gland. We have 26 billion of people with clear brain damage, developmental brain damage, which could have been prevented. And we still have approximately 6 million of cretins, this meaning mentally and physically impaired individuals worldwide. We could prevent it by increasing iodine in the table salt and in the food. It is not done yet, but it's necessary to do that. This is a first sign, increase of thyroid gland, goiter development, that the thyroid tries to compete. You can prevent these diseases and their sequelae later on with adequate iodine intake. And to give you a news here from local India, I just recently found this paper from 2012. What is the situation in India? There has been countrywide universal salt iodination introduced over the last 2 decades, and they made an analysis covering almost 39,000 children. Still 15% have a goiter, have inadequate thyroid function, and even hypothyroidism was there. So in spite of nationwide universal salt iodination, we still have the problem here in India. So it's not enough to have table iodine, salt. You need other sources of iodine to get adequate intake, and everybody theoretically should use it, which is not the case. Now, what does it mean for children? Another publication from India. You see that already 10–15% have a goiter, enlarged thyroid gland, so they are at risk of thyroid malfunction. And then see what's happening with puberty, 13 to 16 years, female girls will get higher incidence. So there is a sex component in thyroid dysfunction, which is still not understood and very clear. And if you look at urinary iodine excretion here in Bangalore study here in the city area, 300 first trimester pregnant women, and unfortunately, these 2 groups have inadequate iodine excretion in their urine. That means their kids will have less chances to get an adequate IQ, 10–15 points. This could be prevented, of course. Now, let's go to the thyroid hormones. How do they act? How are they? The peculiar thing with thyroid hormones is they are aromatic compounds derived from tyrosine, but they have these huge iodine atoms, 4 in thyroxine, 3 in T3. And if you see then, you can change one iodine, take it out, via 5' deiodination here, and you end up with a biological hormone. Though this is probably not active, is a pro-hormone, and this is the active hormone. So the body has to make out of the precursor another active hormone form, and this is done by the deiodinases, and lo and behold, these enzymes are selenium dependent. So you have a second selenium dependency, not only in the synthesis of the gland, but at the step of activation. You cannot make deiodinase without selenium, so you cannot make adequate T3 production for the human, for the animal, with lack of deiodinases. Now, deiodinases are selenoproteins, so the thyroid gland secretes thyroxine, approximately 80%, but it also secretes already T3, and T3 is then thermogenic and metabolically active, and the deiodinases will remove from the precursor this iodine atom in a reductive deiodination to make the active hormone T3, which then can act. And then it can continue, and that's new. You can continue further, take another iodine away, and you end up with 3,5-deiodothyronine, which does not have iodine atoms. And this is a new thyroid hormone which has activity on its own. It does not act primarily via the nuclear receptors. It acts mainly on mitochondria and activates mitochondria to increase oxygen consumption, basal metabolic rate, and so on. So there is an independent thermogenic acting of a metabolite of T3, which is currently studied and analyzed. And big surprise, a few years ago, there is a 4th hormone, which is called 3T1AM. I give you the structure. 3 iodines have been removed, 1 is left, and in the side chain, a decarboxylation took place. So we have a biogenic amine with an iodine in it. We do not know yet how this is made. We know that it is circulating in our blood in concentrations as high as this one. And now look at the person. If you give thyroxine, you decrease body temperature. This has not been done in human, but in animal experiment, reversibly by 7 to 8 degrees. So this is a biogenic cryogen. Very interesting for the intensive care. You could send somebody to the moon by cooling him down or something like it. A few weeks ago, we were able to analyze the first deiodinase catalytic structure. Not the whole enzyme, but the active site. So now, from the first of the 3 deiodinases, we know how in the active site thyroxine is encouraged to be deiodinated, and we are now having a target, and hopefully also for the other 2, deiodinase 1 and 2, to get new information, and in cooperation then with dedicated chemists like Dr. Muge's group to design new targets and site-directed reagents. Now, last part, how does thyroid hormone come really to the cell? You have an amino acid which is charged, and I guess if you open your textbooks, many of your textbooks say thyroid hormones are hydrophobic, lipophilic, and they will diffuse through membranes because they are lipophilic. This is nonsense because they are designed from an amino acid, and every amino acid is charged. And thyroxine even has one more charge, a third charge at the phenolic group. So you need cell membrane transport or facilitator systems to get the hormone into the cell and to get the hormone out of the thyroid gland into the blood circulation. And it took very, very long time, and it took a patient to convince medical community, textbook writers, and textbook copiers to accept that thyroid hormones are transported as amino acids to plasma membrane, because the first mutation was identified in 2004 by my current dean of the medical faculty, Annette Grüters, because she found the first transporter mutation. So what is happening here, this is the textbook scheme. You see, thyroid hormone is made, it's transported in the blood, bound to binding, and then it diffuses through a lipid membrane, and then it goes into nucleus where it acts with nuclear reaction. This is not true, because we have transporters in the cell membrane, and they are limiting, and they are cell-specific. And we are currently probably having 15 different transporters for thyroid hormones, which are expressed in development, tissue, and time-specific manner. Not every cell has the same repertoire. So they have to get in, they have to be transported. All the iodinases are inside the cell, and then you can get T3, which is acting with the nuclear receptors. And here you see the structure of one of these transporters, which have been classified, not yet characterized. And this is a patient, the first patient identified. Muscle hypotonia, developmental retardations, no speech development, low body muscle mass, etc. Very fatal phenotype. The patient has spontaneous dystonic movements, so cannot really react, needs to be fed. Severe disease, and now we have a homology model of this transporter, which is rather similar to the T3 transporter, so we can modify with our colleagues together in this priority program new targets also for the transporter for the nuclear receptors. So this kind of summarizes what's happening. You have thyroxine coming to the cell, you have T3 coming to the cell, and then you have in the cell transporters, intracellular deiodinases, and then the receptors which modify gene expression up or down. Now, the structure of the receptors has been studied and shown quite diligently, and we know what they are really, where they are binding their ligand. And I will just mention here, because I mentioned we need 4 essential trace elements. To make the receptor work, you need zinc fingers in these receptors. These are these helical structure coordinating zinc. And without zinc, you cannot really interact with the DNA. So here you get the 4th trace element in the thyroid hormone metabolism, action, and synthesis. Now, we have development, and I come to the end here, now a system to study whether any reagent will interfere with thyroid hormone action at the nuclear receptor. So we developed something called a luciferase reporter assay. I don't go into detail. The principle is you have the receptor in the cell, you deliver the ligand, and the ligand competes with receptor action, and the receptor activates gene expression, a light-emitting gene, luciferase, which you can easily measure in a high-throughput system. And we use this system, Peter Hoffman especially, to study many, many compounds. And what are the compounds? These are chemicals from our environment. Synthetic chemicals from agriculture, pharmaceuticals, plant products, natural compounds. And we screened them whether they would interfere with that action. And they do. We found only in a first series of 23 compounds, only 2 had no effect on the nuclear T3 receptor gene expression. This series has now been expanded to more than 200 compounds, and approximately, I would say, 70% of these compounds interfere in vitro in the cell system with T3 action at the receptor. And this is not a good news. We are concerned that many of the endocrine active compounds, and here you see the production rates of chemicals over the last years, since 1940, they are not well characterized, they are used, they are distributed in our environment, and we are afraid that they not only affect the reproductive function, but also the thyroid function. And if you look at the 30,000 of the endocrine disruptors, which are currently screened with an EU program called REACH for their potential interaction, there is a lot of work to come. So we are needing new ligands for the binding and transport proteins, and I will come to my summary. Adequate thyroid hormone synthesis and secretion is crucially essential for human development, especially brain development. We need 4 trace elements for adequate function. Thyroid hormones are also affecting growth and metabolic function, energy metabolism, and now we have learned, and this is not the good news, we have to distinguish, that many of these hormone synthesis, transport, metabolism, and action processes can be affected by endocrine active compounds, endocrine disruptors. And we have to study that, whether this will interfere. And we need also new, let's say, ligands, targeted ligands, to interfere with hyperthyroidism. And I would like now to thank you for listening. Many thanks to the Humboldt Kolleg and Professor Mugash for the invitation and organization. My interdisciplinary team at the Institute, and especially my 2 group leaders, Lutz Schomburg, Ulrich Schweitzer, and now Eva Karwert and Kostya Renko, and the whole community for supporting this research over the last years. And this was, of course, only possible with a lot of funding from different sources, German, EU, and cancer foundations. And I thank you for your attendance.
So we can afford to have a couple of questions if you are interested. Are there any queries, comments on this lecture? Yes, please.
Contrast with the kind of diffusion mechanism which can lead to slow diffusion of the thyroid cells. How was that, like, how did the pathway advance? What is the marker for the pathway?
We do not know that yet. So currently there are 3 of them studied, MCT8, organic ion transporter, and LAT2. And there is not yet much known on their regulation by physiological and pathophysiological processes, and what we currently try to identify to find inhibitors for these individual transporters so that we can study them independently. And based on the mutations of this MCT8, there are many patients, meanwhile, more than 100 families identified who have a MCT8 mutation with a very different phenotype. We try to learn most about MCT8. So we know that export of the hormone from the thyroid gland is dominantly affected by MCT8 export, that the transport of thyroid hormone from endothelial cells into the tissue depends on OATP mainly, and that some of the brain areas are regulated via LAT2 in terms of their T3 access. But the real chemistry is not yet.
So you mean to say that without the presence of this active transporter, the hormone cannot enter into the cell? Is that the way?
That's what we think. Because it's not plausible that a protected hormone, which is bound to binding proteins, is just flip-flopping through the membranes in a charged way. I think this is really difficult for me to imagine, and for many others. So if it would go into the membrane, it would get stuck because it's rather tall. So if it's in the membrane, it doesn't go out. So what the body actually has to do to prevent that hormone is kind of concentrated in a phospholipid membrane. It has to prevent that. Yeah. So we need transporter or facilitators. Some of them might be energy dependent, some may be sodium dependent, and others are just facilitators. The hormone transporter modification takes place when we analyze cells. By Western blot we find two forms of the modification, but we are not sure yet whether it's phosphorylation, glycosylation, or another possibility, addition of a lipid anchor or something like that. So this has not been studied. But the nuclear receptor, T3 receptors, there are two of them, they are clearly regulated via phosphorylation and dephosphorylation.
Okay, one more question. You mentioned about this iodine is not electrolytic process, iodine transport. Iodine
Yeah, uptake. Yeah.
is not an electrolytic process.
No, it's electrogenic, and it's driven by this by the gradient of the sodium-potassium ATPase. So there is one iodine with two sodium cations, and the driving force is the sodium-potassium ATPase, which gives the counteraction to the gradient, the sodium gradient.
Are the transporter for this iodine coupled to sodium-potassium ATPase, or is it the sodium-potassium ATPase itself which does the transport?
Yeah. No, this is the only one known. There is on the apical membrane, which is facing the colloid, there is a second iodide channel, which is called pendrin, but this is not linked to any gradient. So the efflux into the colloid is differently regulated than the uptake into the cell. So there are two different systems. The second one is not yet well studied.
Session. This will be given by Dr. Sabik Maiti from the Institute of Genomics and Integrative Biology from Delhi. He had his early education at Jadavpur University in Calcutta, and he did his PhD from the Indian Institute of Chemical Technology in Hyderabad. Subsequently, he went to the United States and France for his postdoctoral training, came back, joined IGAB again as a staff scientist, where he has been for a long time now. And his main area of interest are generally the so-called chemical biology of nucleic acids. Today he will talk to us about interfering with interference, targeting microRNA with small molecule. Dr. Maiti, please.